It's my very great pleasure to welcome you to the annual prize lecture in Life and Medical Sciences, so Life and Medical Sciences 2020. And I have to say I'm delighted that we've decided to stage the lecture, even though we can't all get together in the usual way. So we're doing all of this by Zoom. My experience of this is it is quite possible to create quite an atmosphere around Zoom when you've got great participants, as we have this afternoon. Just a little bit of background on the lecture. This is a lecture that's been running since 1997. And as I explained to Anne Graybiel just now, this is our prime lecture of the year at UCL. And so it's a very great pleasure and to have you participating. The way this works is that we ask all of our staff and our students to make nominations, make suggestions for nominations, and then we have a committee sift through. And I can assure you, Anne, it's almost as exacting as the Nobel Committee. If you get through this and get selected for this award, it's because you're a very, very distinguished scientist. So congratulations on that. And if I could just say a few words of introduction about Anne herself. She is Institute Professor at MIT, where she's an investigator in the McGovern Institute for Brain Research and the Department of Brain and Cognitive Sciences. She has been elected to the National Academy of Sciences and the National Academy of Medicine. Her achievements is a great long list, but I'm just going to pick out two which are particularly noteworthy, and they are the National Medal of Science for the USA, which was awarded in 2001, and the Kavli Prize awarded in 2012. Anne and her laboratory are actively investigating neural circuits related to basal ganglia, and without doubt, she has transformed the field of understanding decision making. We tend to make many decisions about what to do based on how we evaluate the costs and benefits of our potential actions. And situations like the one that we're all facing with COVID-19 tend to bring such cost-benefit decision-making to the fore. The reason for this being that as a society, we're having to think through actions which previously would have had little impact. So the lecture today will highlight the neuroscience research that focuses on circuits that are called to action during challenging decision making. I'm going to hand over very shortly to Anne. But before I do so, can I remind you that you can ask questions and you can do that via the additional system called Slido. And the event code for that is UCL capital letters prize with capital P 2020, all one word strung together. So if you have questions, please do ask them. And Professor David Lomas, Vice Provost for Health, is going to run the question session after Anne's lectures. Without further ado, I will ask Anne to give her annual prize lecture, Making Decisions Based on Value, Mood States and Brain Circuits. Anne, over to you.
I want to welcome you all to our home outside of Boston. I want to thank you for that invitation. I'm actually in our dining room as I speak right now. And I hope that, although for many of you who are listening, this has been a very unsettling time, profoundly so. And although the lecture is being given, of course, in a virtual format, you will make it so that we can think together for a few minutes about a very deep question in neuroscience. The issue that I'd like to talk with you about is kind of shown here, which is, to me, almost like a miracle, which is, how can we intend to do something, decide to do something, and then actually do it? We know that Motivation is a very big part of this that can color our decisions and, of course, our actions. And we know that when we do make a decision, this can influence not only movement, but also thought. But we also know that our moods change, our brain states, our mind states change. And these changes can affect this whole process. And it's come as a real surprise that the deep lying parts of the brain, the basal ganglia, which I'm showing you here in a very early PET scan of a human brain, very much a surprise that this deep brain can be so deeply involved in this process. These structures have critical roles, it turns out, and I'm showing you here our crown and glory, the neocortex, our thinking cap, and here deep down in pink because this was one of actually a very early fluorodopa scan, but just the tip of the striatum. And it is this core issue about how these regions are influencing our states of mind and mood and action that I want to talk about. Now we know that if these loops somehow are broken, then disorders occur, extrapyramidal motor disorders, for example, Huntington's, Parkinson's disease. And we know also, and increasingly over every single year, we know more deeply that neuropsychiatric disorders can also become involved. Obsessive compulsive disorder, aspects of ASD, anxiety disorders, depression. And we increasingly are learning also that these circuits affect, and their disorder affects how well we learn, our tendency toward repetitive behaviors and addiction, how we handle stress. So the view that the field is coming to is that much of what I'm talking about here depends on this kind of circuit. In this cartoon, the neocortex is shown at the top, and here are the basal ganglia, here the striatum, the largest part, and I will talk about it a lot. And you can see that there are pathways, circuits in the brain that lead out from the basal ganglia, and they're thought to both help us select what to do, but also they're thought to project down, they're well known to project down toward motor pattern generators in the brainstem and spinal cord, which have been active under the influence of these pathways, it's thought, for a long time during evolution. I show you here probably at over 150 million years. But there is another set of pathways here involving the neocortex and the basal ganglia, and also involving this reinforcement circuit. I show you here the substantia nigra with the dopamine fibers coming up to the striatum, carrying signals, for example, related to reinforcing actions, helping the selection. And this other set of pathways are of particular interest to us because they start out in regions of the neocortex that are known clinically to be related to mood, affect. They're disordered in, for example, PTSD and a number of the disorders I mentioned. And what you can see is that this pathway leads down this circuit toward the striatum to special zones. And these zones have the special attribute of being able to project directly to the very dopamine-containing neurons that send, in turn, modulating inputs up to the striatum. And indirectly, the pathway can get to the serotonin system, for example, famous for SSRIs, which in turn can have heavy influence on our moods. So I do, though, have to start with an old finding about this circuit. I'm going to tell you almost all very new things, including some not out in publications. But here is what I want to show you. You're looking at a cross section, a thin slice through the striatum of a human, striatum. Here the caudate nucleus, the putamen, here internal capsule. And what I hope you'll see is that there is kind of a dark, matrix-like coloring, and then these little pale zones here. I've got my pointer on one right here and wiggling it around a little bit. This section was stained for a cholinergic marker to mark the cholinergic system that works together with the dopamine system in coordinating aspects of our behavioral control, and is deeply involved in the Parkinsonian patients as well. And so this shows you that there's different chemical constituents of these little zones. I came to call them striosomes for striatal bodies. And you can see the words down here, the bottom. But it turns out that almost all of the neurotransmitters that we know of in the striatum respect this boundary. So either they're higher in the striosomes or in the matrix. The connectivity is different. The development of these striosomes and matrix are different. It's a very striking system. So we knew this from a long time ago, but we had absolutely no idea of anything to do with the possible function of all of this until this happened. And Frank Eblin was a postdoc coming from Germany, worked in the lab. We decided to look at the frontal lobe connections to the striatum, hunting for a hint that one or two regions might particularly project into those special regions. And what we found, you're looking at a medial view of the brain, a non-human primate brain. And what you can see is that of all the different regions we injected, shown in red and blue, just this red region, which is just in front of the corpus callosum, is a genus, so we call it pregenual anterior cingulate. Only that region was found to project predominantly to striosomes. This quickly became highly alerting because of clinical work. People were doing more and more brain imaging after that and found this zone lit up in people with depression, major depression. Here, cocaine addicts, there it is again. And here it is an amazing example. This is the brain of a migrant who came to Germany and was one of many of the migrants who suffered severe anxiety because of this was their first experience in an urban environment. So we were, of course, very excited and elated, but to have some link to function, but we had no idea of how extensive the system was. And so now I want to give you an idea of that by showing you a mouse brain. This is the front of the brain, actually olfactory front. Here's the back of the brain. And in red, you can see some of the striatum. And it's red because we engineered this mouse. We made the cells that project out of the striatum that mainly lie in striosomes. We made those cells have a red tag. So when I start this clarity video, which I can do thanks to the genius of Hong Chung, who invented this method, clarity method, you'll see, and I'll stop it again, we're just about to zoom out. You'll see here red cells, and you'll notice that they are clustered together, those little sparkles, and you'll see that the clusters sometimes converge. The striosomes actually form a labyrinth that runs through the striatum. And I hope you get some feeling that this is a huge, huge part of the striatum. So it's at about this point that Kenichi Yamamori came to the lab. And we decided to tackle the following question. Okay, we have some idea that these special zones of cortex related, let's call it to mood and affect, project down, and some of them project especially to striosomes. We know the dopamine is there. We know they're the output pathways. And so what does that circuit do to our decisions or our selections of what to do? And if we're to tackle that question, what kind of decision making should we study? And so here's what we decided to do. We decided to look at what clinicians call approach avoidance decision making or approach avoidance conflict decision making. And the reason that we chose this is that in the clinic, it is been greatly studied, in fact, that this kind of approach avoidance decision making, which induces conflict, you're looking at something that you're going to have to choose. It's got good and it's got bad aspects, and you're going to essentially have to take the bad in order to get the good, or reject the good in order to avoid the bad. So this kind of decision making is highly vulnerable in individuals with problems with anxiety and depression. And the other reason we chose it is shown here. This is a highly evolutionarily conserved kind of behavior. You can see him looking this way, trying to decide whether to go forward, to approach, or to run away. And you can see that all his friends have already made the decision. And so what Ken and I could do by having this set up is to actually get to numbers, which is what we wanted. We could make a plot of how much cost, which in our case to the animals was a gentle puff to the face, air puff, which we, like other animals, find irritating. And the benefit was food, liquid food. And we could just plot their choices. Here, there are a lot of costs they're avoiding. Here, a lot of benefit, a lot of food they're choosing it to approach. So we could get a decision boundary. That's what we needed, because then we could go to that blue zone that tends to project preferentially to striosomes, and we could microstimulate it and ask whether we could change that decision boundary. Did we move toward approach or to avoid? So we did that, and here is the setup, microstimulating that zone. And what we found, to our interest, was that we certainly could change the decision-making of those animals in a reliable way. It increased their avoidance when the stimulation was on. And when we turned it off, the effect would slowly go away, or it could even last when we were doing this in the striatum. So what we essentially found is that at the head end of the circuit I'm talking about, this manipulation essentially made the animals think that what was coming was worse than it really was going to be, or that they would have thought it to be prior to that stimulation. This effect was blocked by anxiolytics, for example, by diazepam dose-dependently. There was no effect at all in the approach control task. We always carried a control task along. And this made us then think, have we found some form of proxy for what in people we would call pessimism, for making pessimistic decisions? It's going to be really bad, or for even anxiety and depression. And in the introduction, you mentioned that this time is a very a time of vulnerability for many of us, and it's a special, shall I say, we feel especially strongly that we need to study this very carefully. In fact, we've done, through collaboration with the laboratory of Diego Pizzagalli at McLean Hospital outside of Boston, we've collaborated in the first human experiment. And I'm showing you now a human brain. Here is that very similar region in front of the genu, which it turns out when the subjects, these are human subjects, are given as as much the same protocol as our animals had, even down to the same stimuli, but instead of juice, they get happy pictures, and instead of air puff, they get unhappy pictures. And it turned out that sure enough, this region was highly correlated with how aversive they found the offers, and in fact, with how much, in fact, they avoided. So this encouraged us to go on, but we knew we had to get somehow at a cellular level, a circuit level. And so we've tried to do that, and we've done that partly through the mouse engineering. And I'm showing you here with this zoom how we've here for you zoomed in on a striosome, which has been engineered. And how we did this, oops, I'm trying to go forward. It doesn't want me to, it likes to see that again and again. I'm trying hard, ladies and gentlemen. There we go. It is pretty. The reason that I'm showing you that is that we wanted to get permanent tags on neurons so we could use mice in a very parsimonious way. And here are the facts that led us to be able to do it. It turns out that the differentiation between striosomes and matrix occurs remarkably early during development. In fact, it occurs within the germinal epithelium. And I'm showing you here's the lateral ventricle, that germinal epithelium that's going to produce the striatum. And it turns out that the striosomes come from what's called apical progenitor cells. This is work that I did in collaboration with Josh Huang's lab, and now we're working on it in our own lab. Well, all of that was in our lab too, but anyway, here are those apical progenitors. An early tagging with tamoxifen lets us keep a permanent tag so we can see that B cells grow up to be B cells. In sharp contrast, here's what happens when we do that tagging a little later. Now we predominantly label projection neurons in the matrix. You can see they're projection neurons because you can see that these are spiny. So this means that, to me, something I find quite profound, that this organization has been engrafted in our genome to the level that it's can be recognized very early. And also we can use this kind of tagging in our experiments. And so here's what we did. We decided, and this project was led by Alexander Friedman with a number of other individuals I'll mention at the end. What we decided to do is to set up kind of simple mouse, kind of rodent version, binary version of a cost-benefit task. So look here. Here you can see in the T-maze, this is a reward of pure chocolate milk. Mice love pure chocolate milk. But this is paired with very, very bright light shining right down on that little cup where they can get the milk, and this is very, very aversive to them. So a regular mouse will run away toward the other end. But the way it's set up at this end, although the light is dim, the chocolate milk is very, very dilute. So we actually made dilution curves. We measured lux. We did parametric studies to work this all out. But the upshot is that mice avoid bright light despite obtaining dilute chocolate milk. We had we had 4 other control tasks where chocolate, this was light. We had reversed cost benefit, so dim light goes with pure chocolate. And the reason I mention this is that we found something remarkable, which is that the striosomal circuit became active only in the approach avoidance version, not in all these other versions. And I want to show you that here. We were recording with microelectrodes, tetrodes, in both the neocortex and down here in the striatum. And what we found was that when animals were performing this experiment, the prefrontal cortex was on, cells were firing. These were usually identified antidromically as the ones projecting down toward the striosomes. And here, the striosomes, when we recorded from them, they were off, even though this signal was coming down from the neocortex. This was very, very surprising to us. So we decided, well, we'll manipulate this system. And so we did. What we did was we used optogenetics to slow down, almost turn off, that input from the neocortex. And what we found was now the animals were running the other way. Most of the time they were running to the aversive light. They're not avoiding it. And moreover, we found that now the inhibitory interneurons were off, striosomes were on. And we had done this ourselves by tuning down the neocortex. So with this manipulation, what we actually found, and I won't go into the details, is that this is all a matter of timing. If the inhibition, that the normal inhibition is so rapid that it can basically outrun the cortical pathway down to the striosomes, cuts it right off. But if we slowed down that pathway, then what happened was the inhibition wasn't quite enough to turn off the striosomes, and they ran in this really crazy way. So the situation we found at that point was, all right, the striosomes are on, mice do strange things. For example, in this experiment, they approach what is to them aversive. But if the striosomes are off, then they seem to behave normally. This is what brought us to ask the question whether we're dealing with some kind of switching circuit. Could it be that the striosomes are able to turn on and off a large network of circuits to bring about this behavior? In other words, have we actually induced some kind of state change in the brain? So the question, of course, is whether that's fine, it's an experiment, right? But could this happen through experience where we aren't manipulating the brain? So we decided we had to think of some kind of experience that would change, would be strong enough to likely induce a state change, but not too strong. And so what we decided to do was to use a protocol. It's called mild chronic stress. And what this involved was putting a mouse into a little cloth sack so he couldn't wiggle around much. They love to wiggle. And keeping them in the sack for 1 hour a day, just 1 hour, every day for 2 weeks. So they're just, for about an hour, they're not wiggling around too much. I think parents of children will understand what I'm talking about. And then we would take these mice who had been mildly stressed and put them into the experiment. And they actually had done the experiment. They knew how to do it. Later, I'll tell you. But anyway, here is the no stress baseline. These are mice that are simply doing the normal thing. They're avoiding the bright light. And I'd like to show you what stunned us, which is that with pre-exposure to stress, these mice began running toward the bright light. We had not touched the brains. We hadn't done anything to the mice except give them that 2 weeks, 1 hour a day in a gently held little sack. We found then that the striosomes were on, the inhibitory interneurons were off, and this showed us And we really were stunned that experience alone can switch this circuit dynamics. So that asked, they say if you find something, then what you really find is another question. And so it was with us, because then we wanted to know whether we could reverse the effects of the prior chronic stress. And so what we did then was, I'm showing you in the cartoon, what we did was to go down and excite the interneurons, which had been inhibited. And sure enough, what we found was under those conditions, the pre-stressed animals, instead of running to the bright light, ran normally. Found this in many, many, many mice. So what we then could say is that we were restoring dynamics, essentially the temporal dynamics of excitation and inhibition in this circuit. And if we did that properly, we could take away the effects of chronic stress. We were heartened by that, but we also were very mindful now that this circuit is extremely vulnerable. Even slight changes that could be produced either endogenously or by experience could destabilize it. And maybe that in the end will be part of the answer to why, insofar as the circuit is involved in mental state changes that are not good, why that might happen. Now, the question of experience and state change and mental state, there is no better example maybe of a state change than what happens to us as we learn. And so we decided then that we would see whether this value evaluation system itself would change with learning if we had a task which engaged value judgments. And we ended up using what's called discrimination related to value. So I would like to think with you about us. Okay, so we're 35, maybe robust, 50 prime time of life, 60 terrific, and then time goes by. And what we decided to do was to try to mimic those ages by choosing mice at ages estimated to be roughly similar to those of people at these various times. So that turns out to be 6 months, 9 months, and 12 months or over. So to look at the effects of aging, we were very interested to see how the engagement, the motivation of these mice would change with age, because the system was so heavily dedicated to modifying motivation. So we set up a very simple task. Again, it involved a reward. And this time, the cost is a light again. And one tone meant the more the mouse licks, the more reward he gets. The other tone, the fewer he licks, fewer times he licks, the less the light. And so I have very good news. And the good news is that if we make a graph showing you the young, they're not that young, but they're, let's put pretty young, the middle and the very old animals, and we look at the percent of mice that learned across these different ages, essentially everybody could learn. We were heartened. We did, however, then find that if we looked at the early stage of that training, when they were first couple hundred trials, when they're just beginning to learn, then it was fine for the young animals. But by the time we looked at the older animals, they had quite a serious problem with just getting into the task, getting going, doing it. In this case, licking more, licking less, depending on the tones. But another bit of good news is that for the older ones, just if they kept at it, eventually then, and here you see engagement late, far, they could recover, they could do it. But this was looking now after many trials. But this gave us the idea that we might be able to find a signal, perhaps in the striosomal system, that related to this defect. And we did. What we did was we took animals in these groups, in the young and middle group, and we looked at their striosomal signals. By now we're using a photometry in so-called striosome mice, red cell tagged striosome mice, in effect. And if we got signals, and we were looking at the engagement state of the animals from low to high, and you can see that the more engaged the animals were, the more the striosomal system was on. This was very exciting to us because the correlations were really high. They were unmistakable. So again, we can learn across, or at least mice can, across a large range of ages into old age. The engagement, especially getting going, declines, but maybe that's reparable, we wondered, because we found this neural correlate of the striosomal activity. And so we asked whether we could increase the engagement by changing that striosomal activity, namely by increasing it. And the way we did that was by using dreads, excitatory dreads, to excite the striosomal circuit. And we then we found that with increase in the striosomal signal, we found an increase in the engagement of the animals. We did this through looking at different doses from low to high and homed in on ranges for each animal. It took a very long time to do, ranges in which effects were found in the brain. And if they were found in the brain, we could then make the correlation with the behavior. So we find then that we can modulate this early kind of get up and go. We also did experiments where we used inhibitory dreads. We could diminish engagement, but that's not what we wanted to do. So now I would like to wonder with you whether this engagement itself could be a proxy for motivation. It seems for all the world kind of like a get up and go kind of behavior. When you're young, if a little child, just anything he'll run out and do. But for someone getting older, they may have to think, I'll go do that. So with this, I'd like to mention one final point, which I brought up early, and it's extremely important. And that is that this system that I've been talking about is the one that has the special privilege of projecting straight to the dopamine-containing neurons. This isn't the only input to the dopamine-containing neurons by any means, but as I'll show you, the input that it has is extremely strong. And I'm showing you a miniature picture there, but here is a blow-up of a thin section through the brain of a mouse. It's an engineered mouse, engineered so that the dopamine-containing neurons of the substantia nigra, which you see here, are in this pinkish color. Here's the cerebral peduncle, and this is outside of the brain. And here, these green fibers, these actually come from striosomes. That's another bit of engineering, mouse engineering. And what they do is they crawl up the dendrites, the receiving branches of these clusters of neurons in dopamine-containing neurons in the striatum. And I want to show you, actually, this is one of our first, it's still my favorite picture of these remarkable, remarkable arrangements. It's like a bouquet of roses. Here you can see the cell bodies of the dopamine-containing neurons. They're now red in this image, like roses. And here's the stem of the bouquet. You can see the dendrites of the cluster. They twist around each other. It's most remarkable. So you have 10, 20, 30 dendrites twisted around one another. These are the receiving poles, and up come, you can see some in green, the striosomal fibers coming down all the way from the striatum. They're twisted around the twisted dendrites, all forming a dendron. And so we named these Striosome-Dendron Bouquets. Now I'm very happy to tell you that labs that do slice work are finding that they can fully block the activity, the electrical activity of these dopamine-containing neurons in a slice prep when they microstimulate the striosomal fibers. This is now true of several labs. I wish I had more time to talk about this with you. But the exciting point then is it's perfectly possible that the dynamics I've been telling you about for the upper part of this corticostriatal system, that those dynamics can play down on the dopamine-containing system, and they can literally turn on or off or modulate in another way the dopamine-containing neurons themselves. This could be a very powerful control system or modulating circuit, which might be a circuit for changing mood or our mental state. This is something we're thinking about, and I know that we cannot say for sure that that is so, but what I can say is that everything that we have found out and others have found out about this circuit suggests that it has power, that it can affect the mental side of our lives as well as the physical and motor side of our lives. And it's come as a new and quite interesting finding or set of findings that actually neurons in the system, some of them are encrusted with receptors for psychoactive ligands, compounds. For example, serotonin receptors are extremely strongly enriched in striosomes. So are opioid receptors. That's been known for years, the mu1s. And here we found also, in addition to the serotonin, we found that cannabis receptors, endocannabinoid receptors, CB1 receptors, are encrusted on the dendrons of the dopamine dendron bouquets. So it's a powerful system. We want to think that somehow nature, long ago, invented a very simple circuit for helping us run away from something bad or run towards something good. But with evolution, this system has played up to our forebrains, to regions that are not just controlling running away or running toward, but to controlling the moods that we have, the brain states we have, how happy or pessimistic we are. And that turns me back to the disorders that I brought up early in this talk, Parkinson's and Huntington's dystonias, and many of those, that many of these disorders have a cognitive and emotional aspect as increasingly is being found. For example, it's now thought that in Huntington's disease, so-called manifest Huntington has been thought of as when the motor disorder can be diagnosed, but up to about 10 years before that, people may have really terrible mood disorder problems. And so these are not any longer called motor or just extrapyramidal motor, but motor plus disorders. And here this range of disorders in the biopsychiatric realm and our own behaviors, including addictions, which I think now, seeing all those receptors, we must think actively about new ways to rehabilitate individuals with these problems. But overall, that brings me to say to you that despite the fact that we are in tough times right now in a number of parts of the world, the work in neuroscience has reached a point where almost certainly pure science can lead to translation to medicine. And it is profoundly important that this time has come. It is a very magical time. And it makes me all the more grateful that you have invited me to give this very special lecture. And so I would like to end by mentioning the names of people, some of whom I have mentioned, who have been important in this work. Ken Kenichi and Satoko Amamori, Alexander Friedman, Emily Hicksta, much of the mouse work along with their collaborators here, Joe Crittenden and Ayano Matsushima for some of the molecular work. And then our collaborators here, the tagging of the germinal epithelial cells, the expansion microscopy, here the work on cannabis, and we have received reagents from one very generous Carl Deiseroth. We've been funded, as I can show you here, by these institutions, and I do want to say, just as I say that pure research is important and can lead, not with hundreds of years lag, to important clinical help. Not only that, I also want to say that for any work in our field or others in science, we can do this only because there are institutions and agencies and foundations that help us. Thank you very much.
And there would now be a huge round of applause from the audience, and they're out there applauding, but unfortunately we can't hear them. So can I just say that's absolutely fabulous, truly, truly wonderful talk. Thank you very much appreciated. And to our audience, if I may, just to remind you that you can pose questions to Anne, and I'll try and read them out. If you go to Slido, sli.do, and enter the code UCL Prize 22, all one word, with UCL and the P of prize being capitals, and then the rest being lowercase. So UCL Prize 22, one word on Slido, and I'll try my best to read out the questions. So Anne, if I can pick up on the last point about being in strange times, the first question actually came on the effect of the pandemic and the changes that's brought in people in terms of their decision making.
Yes, so it is a time when many people have noticed that they have trouble making decisions, that they're more hesitant about doing one thing or another, or worrying, do I dare go out? Can I, after all these months, go to a restaurant? Or is it safe to have my child at a school? And so a general state of uncertainty or anxiety certainly is around for I think many people in different kinds of life experience. And what I think people can do is, if they can possibly bring themselves to recognize that, to say, look, times it's reasonable. Actually, it's reasonable to be stressed out. It's reasonable to have kind of my brain state different and affected maybe badly, or to have people around me being irritable or in some way behaving abnormally. This is part of our normal behavior in an abnormal time. And if people can think that, I truly believe that that can be in and of itself rather therapeutic for people. We often, as you mentioned, we so often do things almost without thinking. Our lab has studied habit learning for many, many years, and it's absolutely amazing that when we learn a habit and we really, really learn it, it turns out that the habit circuits, and we've done this with physiology and with optogenetics and many methods, turns out that these brain circuits, they change too. So they change, and they change both at the level of the neocortex and at the level of the striatum, the two places that we've looked. The other places are changing. And what's amazing is that they begin to change, and then when in animal experimental jargon, it's called overlearning. When the habit is really just pushed in by doing things over and over and over again, then the circuits undergo the greatest changes. And if at that point we change the circumstance so that the habit doesn't work, as it were, the neocortex quickly changes its pattern, its habit pattern. It drops it just in a shot, but the striatum does not drop it. The striatum keeps that newly formed pattern of electrical activity in ensembles of its neurons. It just won't budge. And this, I really do think, is why habits are so hard to break. And here we are, and this is talking a long time in one question, but here we are in the midst of this COVID crisis. Yeah. Our habits are absolutely disrupted, and yet it's very jarring to us, probably in part because our striatums are saying, no, I want to do my habit.
Yeah.
Thank you. It's a long answer.
So can I follow on from that and just saying, so are these pathways active in humans? Do we know they're present in humans, and can we modify them in humans to change moods?
Boy, one thing that the striosomes themselves are just beyond the resolution of fMRI or BOLD at the current time. It's really they're getting close, but not yet. So we cannot see them per se. But what we can see is those regions in the neocortex. And I showed you one image from our own collaborative work. There, what I would think we could do is to use the kind of fMRI in which people actually are given a vision of the activity that's going on in their brains. They can see it on the screen, and then they can try to change that activity. And that works quite well, in fact. And so with that training, some changes have been made. It's a very active field of work. I don't think it's been done in this context, but I think it should be tried, because some of the problems that we're talking about are severe enough that they would do with such an effort.
Can I ask you an interesting question, actually, that's coming from the audience? Do you think people would be happier if their decisions were made for them?
Oh my!
I thought you'd like that one.
Yeah,
thank you, whoever said to do.
I'm going to get in bad trouble. I think any child over a couple of years old will begin to say, I want to do this.
Yeah.
So this is all normal. Could there be a stage at which, there just is too much oversight on us? I think for many people that absolutely would be true. So I don't think it would be good for all of our decisions to be made for us, because I think actually exercising our brains is a good and fun thing. If we aren't in a terrible situation, it can be fun and kind of mind expanding to do that. So I think we have a natural tendency, I think all all creatures do from probably slime molds on. All creatures do have the tendency to explore. You know, you have to explore your environment until you find the food you want, the fluid you want, the mate you want, and then you exploit. That is to say, you're able to have drink the water or whatever. But even though you're at that nice sweet spot, you're at some nice little spot, you still have to explore some, because maybe things will change or maybe something bad will happen. Mm-hmm. So we're in this sort of explore, exploit brain rhythm, which I think would be a real shame not to have some use of that. So no, I don't think all of our decisions should be made for us.
Great, and thank you. Can I take you to disease now, if I may? So abnormal and insufficient movement, if you like, in terms of Parkinson's and Huntington's disease. You perhaps look on different sides of movement. One difficult initiating movement, one, if you like, too much movement, and a career-form movement. What does your system tell us about that? And does it open the avenues for new therapeutic strategies for either of those entities?
This is, of course, this is an enormously important question. I can say a little bit about Huntington's disease, the reason being that two laboratories or sets of researchers have found that striosomes are differentially affected in some Huntington's patients. In the first study done by John Hedreen, who actually I was in the same lab as him during training, John Hedreen found that in a series of postmortem samples, was a crackerjack pathologist, he found that across a series of patients, striosomes were affected earliest of the cells in the striatum. He looked at gliosis. In the second study, was done by the laboratory and clinic of Sir Richard Fall of New Zealand. We also knew each other from a long time ago, and I had the privilege to work with them on postmortem cases. And again, there were some brains in which striosomes were differentially affected, meaning they were little. And these turned out to be from individuals who had suffered mood disorder as their early symptoms. We had 37 cases, which is not enough, but there are new suggestions that from snRNA-seq studies, McConnell here in Boston, that in just a few cases so far, that striosomal mRNAs are heavily affected in the sense that they're diminished greatly relative to those of matrix. And we ourselves have now done a study with using this single cell RNA sequencing method, which gives you single cell resolution. That's what's so wonderful about it. And while we don't find that, first of all, we find there are many, many genes that are particular to striosomes of the D1 type and of the GO pathway, and others that are particular for the no-go or D2 pathway. So there's no doubt that now that the striosomes are entities as defined by their genes, and there are lots of different ones. And in our study, we're still analyzing, but it looks as though some very, very important genes that are striosomal are dysregulated in Huntington's brains and in models thereof. So I think the way to therapy would be to do more of the molecular work, along with everything else that's being done, to try to get better targets for therapy. And I think in the case of Parkinson's disease, there are many, many hints from, including work we've done over the years, that maybe striosomes, well, certainly striosomes in matrix may be differently affected in animal models. The issue is it's very difficult to do in the human brains so far, because the marker that best used to be used was a dopamine marker, and of course that was gone, so that we couldn't, for example, see them anatomically. But again, now that we're beginning to be able to identify these genes, now we can go to the Parkinsonian field, which we do intend absolutely to do, funded, and apply the molecular knowledge to trying to get better targets for therapy. And it's very, very important to do.
Yeah, and thank you. More questions, if I may. So can I take you to the matrix that you mentioned? So based on the findings, would you say the striosomes are the evolutionary older chordate and the matrix evolutionary the newer chordate, or vice versa? And you can flip a coin if you like and answer it either way.
Do you happen to have a coin I can use? You have to flip it all the way across.
Fair enough. That's a good. That's good. Let's.
I can tell you, I think pretty old. I mean, there are dopamine-containing neurons in many supposed current representatives of evolutionarily ancient animals. I always have to be careful how to make that sentence go. There are plenty of dopamine-containing neurons in fruit fly brain, and one of my pastimes is actually to compare the fly brain system to ours, but they certainly have approach and avoidance, but they also, these flies, but they can fly just fine, can't they? And sting and bite. So which one is older? You know, I think how can I put this? I think that in the case of the neocortex, one way of thinking is that early on, we didn't have as many cortical areas as we do now. We have, however you count, we've got 100 cortical areas or more or less. And that early on, maybe what happened was that a given cortical area, let's take a visual area, take primary visual cortex, say it got duplicated by gene duplication. Okay, so just have an extra little piece of cortex. And maybe that survives because, now you can put motion in one cortical zone and not worry about orientation, and put orientation in another, and so on and so on. So that's one idea of evolution of neocortex. And a lot of the neocortex, other than the mood-related areas, is projecting to the matrix. Now, in the case of the germinal epithelium, it's very interesting that those early cells, the apical progenitors, they only have a certain lifespan. They make themselves and throw off, make themselves one and throw off a daughter, and that goes on for a while. But eventually, the cup is empty. But for the basal progenitors that are going to make the matrix cells, they can keep on dividing and dividing and dividing and dividing for many, 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 many times, which leads to the idea that the matrix itself could become large because of that. And that has to go hand in hand with this evolving neocortex, because the inputs are probably required for the whole development of the system. So which is earlier? My guess is if I had to say, I think it would be the striosomal system. You probably gotta see, right? You probably gotta feel some might want to hear, might not, or sense electricity or something, but gee, you better know how to run away from, if you're a little amoeba or something, little paramecium, you still to do something to get out of the way.
And thank you. How can the mice exposed to stress go towards the more aversive stimuli? Wouldn't you expect the opposite?
My dear professor, it is time for you to answer this question. Look, I mean, I honestly, it's a finding, okay? But we have ideas. And one idea is that this system being regulated by all these regulatory molecules like serotonin and dopamine, and them being, I should have added to you the new finding, which will just be coming out in Heartbeat very soon, that when the dopamine does hit receptors, dopamine receptors in the striatum, it's a new finding that the effects are opposite in the striosomes and the matrix. That's quite a cipher, answering your question of why the run to the bright light. But our idea is that with these modulators, if there is some disorder in them, some defect, some change, some timing, some circuit dynamics problem, then maybe overall there's greater impulsivity. Maybe there's greater, to talk about get up and go, maybe there's just a wild kind of get up and go. Maybe our ability to differentiate good and bad decays. We no longer, for example, we don't care so much about cost. And I could tell you something interesting from the work. There are many things I didn't tell you about, but one is that in the experiments on where we did look at cross aging, little kids, they're driven by, they're very sensitive to cost, to good and bad, but they don't care. You know, they just run around and they do all kinds of things. And so it was with the mice. They're very sensitive to cost as well as to reward. But in the older animals, they tend to begin to care much more about reward. So they're much less cost-driven in their behaviors. And so if you think about it and you think about maybe that circuitry is decaying, and I left out a pretty nice slide to show you an example of that eventually will decay, then you could think that the cost, we're just not so sensitive. And in the Huntington's model mice that we ran in other experiments related to this, they just care about the cost. It's a terrible thing. So I think it's a whole change in the teeter-totter of our sensitivity, which is that beta in the equation early on, the beta that gives us some kind of numerical grip on the ratio of benefit to cost. Is that an okay answer?
Yeah, that's very good. And I've got loads of questions. I'm going to just give you one more, if I may, and then let you off the hook at that stage. Otherwise, I think we might keep you all here all evening. So I'm going to go to Frith, actually, who says, "Thank you for an inspiring talk." And I think that's a view that many of us hold. And just, is there a difference between cost avoidance and apathy?
That is just such a good question. And apathy.
Thank you,
Uta. I've thought a lot about the fact that for example, meta studies that were done on patients with damage to the basal ganglia, these are studies that usually are looking at old literature. This is before imaging. So, and what they found was that the single outstanding symptom or symptom complex that they found related to basal ganglia dysfunction was a kind of apathy, anomie, just this kind of like mental fatigue or something. And if you think about avoidance of cost, you can also think of it maybe that way, that you just, you sort of in a way had it. So, okay, here's some more cost. I already have costs in my life. Of course, you could flip it around. If you flip around your question each way, then it's always good to think that way, right? What's one way, what's the other way? If you become very sensitive to cost, like almost hyposthesia, then this can play havoc with the very neurotransmitters I've been talking about and with the whole system. I just think it's a very deep question, and one that we don't have a good enough handle on. And the problem goes back to another of your questions, is how are we gonna exactly hook up everything I'm saying to real-time imaging at the right level of resolution in humans? And then how are we going to use that imaging, and imaging methods are getting better and better and better and better, how we use that, flip it around to be therapeutic? And I do think, I'm firmly in the belief that we're going to have methods that are mildly invasive. In our lab, we're working on, this is the work of Helen Schwert, on tiny little micro probes. They're about the thickness of a baby's hair that can be put into the brain through the method she's created, which is also magical. They hardly damage anything, and they could be used maybe to adjust a circuit, or maybe we can use our own behavior. I am a huge, I do think, I don't want to overdo it, but I do think that better behavioral methods could be developed to help us with some of the problems that we have. And many of us do have problems with mood and brain state. I think almost everybody gets up some days and really happy and I can do anything, and then wake up one day and have absolutely no idea why. You can't figure out a reason, but you just kind of feel down. These are manipulable, I do believe, which is why I was so interested and still am in habit formation, how to get tricks. I'm interested in how we can find tricks to help our brains. Just regular old, normal, run-of-the-mill people who don't have a brain, diagnosed problem, but just would like to have more zest in life, be more up. This, I think, we could perhaps work on even harder, and I know there are lots of methods out there. I think we can have even better ones. But I thank you. I thank you for all the questions, actually. They're wonderful.
And thank you very much indeed. Fabulous question. Great answer to end on, so thank you. Can I apologise to all the people who sent in questions who we didn't have time to read out? They may be in contact you after the lecture, if that's okay, and with extra questions. So can I just finish by saying thank you. It was a great lecture. Thank you for being so tolerant and answering all the questions. And I'm now going to hand over to our President and Provost for the vote of thanks. So thank you very much.
And thank you very much. That was just an outstanding lecture. There are a few things I want to say about it and about you, but I have a series of thank yous to go through, so we'll come back to you shortly. So first of all, thank you to Matt and Sonia for all of the organisation and for pulling all the technology together. We've had a fantastic UCL prize lecture in life and medical sciences for 2020. And I don't think it mattered at all that we weren't on campus. The quality of the science was just brilliant. The team that came together to select the nomination, I want to thank all of them, because I know they will have gone through numerous considerations. They're all members of medicine and life sciences at UCL, and they include Uta Frith, Ibrahim Abubakar, Jill Bates, Mariah Fitzgerald, and Ian Zachary. So to the five of you, thank you for your hard work in selecting this year's lecturer. And what are these big lectures all about? What are we trying to achieve? And I always think it's just wonderful if we can select fantastic role models for our younger scientists, for our students, and for our young scientists who are listening in this evening, and of course for the rest of us too. That role model was very, very evident from this evening's lecture. So first of all, you've been tackling very important science from a very basic set of concepts, a very ancient system. I was blown away by the notion that this system might have been relevant for 150 million years of evolution. So even that, sort of preliminary opening comment grabbed my attention. Other people have used the word inspirational, and I'd like to totally agree with that. Also, I think you've given us a beautiful demonstration of asking simple questions, or apparently simple questions, let's put it that way. And the way in which you introduce each new finding and each new set of experiments with a nice simple question is a real gift. And then the elegance of your experiments. I've turned the bright lights on myself, by the way. I'm expecting the pot of chocolate any moment, but just the way in which you teed those experiments up was fantastic. And then the other things that are very obvious is, your lifelong commitment to this science, and a combination of humility and just sheer brilliance, and great integrity. You just ooze great integrity. So hopefully all of our students and our staff will have picked up on all of those qualities. And the other thing I would note is the wonderful range of techniques that you have used in your science, particularly some of the neuroimaging, both the murine and the human. Some beautiful images. The bouquet of roses I jotted down was also one of my favorites, but also the one you showed of the murine red brain striatum, that complex image fairly early on in the lecture. And of course, very, very topical. How do we make decisions under pressure? All of us have been under intense pressure now for weeks and months on end, and we're all trying to make the best decisions possible in very tricky circumstances. So the poignance of your lecture tonight has been really evident to all of us. So on behalf of UCL, on behalf of everyone in life and medical sciences here at UCL, a huge thank you to you for accepting the invitation and then turning up and giving us a brilliant lecture, albeit with technology and Zoom and everything else that's made it possible. But it's been fantastic, and we are very grateful to you. We wish you well for your science in the future. Much more to come from you that we also could sense. So many congratulations on a brilliant lecture, and thank you from all of us.
Thank you.
It's our absolute pleasure. And when you're in London, when you're next in London, when movement is much freer and we can get back together, it would be a pleasure to invite you into UCL for a coffee and a catch up. Thank you indeed.